Hello, everybody. First, I'd like to thank the organizing committee for inviting me to give a talk here. It's a great pleasure for me to be here, as Tel Aviv University has been a second home for me for many, many years. And I'm a computational biologist at Evogene, and I'm going to talk about the computational discovery of novel pesticides, where pesticides stand for herbicides, insecticides, fungicides, and more. Okay, so first I'm going to give a brief introduction to Evogene. Then I'll talk a little bit about challenges in the industry, in the agrochemical discovery. I'll describe our solution for computational alchem discovery of pesticides, and then summarize. Okay, so Evogene is a leading biotechnology company for the improvement of crop productivity and in performance, addressing the world's increasing demand for food, feed, and fuel. Along the years, we've discovered we have been operating in three areas: in genetic elements, chemical compounds, and microbes. And together with our key world-leading collaborators, seen here, we aim at getting improved seeds, innovative ag chemicals, and novel ag biologicals. And just a few financials. Our main headquarters is located in Israel, and we are publicly traded both in Tel Aviv and Nasdaq. And currently, we are around 180 employees, where 60 of them have PhD. So the idea behind our technology is, behind our development is as follows. First, we have recruited people from diverse disciplines in biology, chemistry, and computer science. Then we curated the data from mainly public resources, but also generated our own proprietary data. This data is then interconnected into currently 4 databases, on top of which we've created 6 platforms seen here, and where I'm going to mainly concentrate on the point for chemical discovery. All our computational discoveries and predictions are validated in our labs, in our greenhouses, and in the field. So what are the main challenges in ag chem discovery? So to identify them, let's first compare the agricultural chemistry with the pharmaceutical industry. So first, our main aim is to improve crop productivity, and this means eliminating weeds or insects. Validation is done in planta or in vivo before in vitro, and this is due to the low pricing. We have very strict environmental fate regulations, and we have to consider the toxicity to mammals and beneficials like honeybees. The product price must be low, and just to give you the feeling, price is measured by dollars per hectare. There is a major lack in target structures, protein structures, and this is due to the fact that this is of relatively low priority to crystallographers. And last, as for activity and functionality of a product, there are no public tools, and therefore we have to develop our own agricultural activity tools, on which I'm going to elaborate in the next few slides. So how we choose to address these challenges, then as for the price, the product must be synthesizable and with high specificity and appropriate stability. As for targets, we have to settle for homology modeling together with molecular dynamic simulations. And then as for ag activity, we have to actually develop our own dedicated machine learning classifiers. Okay, so the overall approach is that first, once a target is discovered in this platform, in the pointer platform, then we run virtual high-throughput screening with PointIt to yield candidate hits, which are then validated in-house. And once hits are confirmed, we run a computational lead optimization, which is then experimentally validated, usually by outsourcing. And then once we get optimized leads, then together with a collaborator, this goes to the phase of development, commercialization, and commercialization by a collaborator. Okay. So how do we address the computational discovery of pesticides? I'm now going to discover PointIt. So the input to the system is as follows. First, first we have the target, and together with the Pharmacophore, which is a virtual representation of the key features, interacting features of the target's binding site. Then together with our own proprietary compound database, which currently comprises of around 150 million compounds, which make more or less 15.5 billion conformers. These are fed into our pipeline, starting with Pharmacophore matching done by Catalyst, followed by rough docking and fine docking, which is done with Glide. And this is the automatic step. And at the end of this automatic step, we are ended with around 10,000 of compounds. And then we have the semi-automatic phase on which I'm going to elaborate soon, that comprises mainly of the ag activity modules and other modules that we have identified. Then the last stage is eyeballing, where a researcher manually selects a compound for the next stage. So all together, we do the virtual screening of more than 150 million compounds in just 5 days per single binding site. So concentrating on the target structure and Pharmacophore, first, I'd like to note a very, very important note that less than 50% of the plant proteome can be homology modeled. And so that's why this is a major constraint for our target discovery platform. And so once we identify a target that can be homology modeled, then we do the homology modeling, and so each one of these boxes that you see here encapsulates a full methodology behind it. So when templates are recognized, we may generate a few 3D models, meaning that it can be an unbound model or a bound model. And from each one of these models, then these are fed into molecular dynamic simulations, where we then cluster all the snapshots of the trajectory, and then pick the significant clusters and we actually pick the representatives from significant clusters, most occupied clusters, and go on into binding site characterization, and then for each such model, we then generate pharmacophore, a couple of pharmacophores. So at the end of this stage, we may end up with many pharmacophores, 20 pharmacophores, and all these together with the model are then fed into the pipeline. As for the compound DB, so the idea behind our proprietary database was that it must have all compounds which have to be which can be synthesizable, and therefore currently there are, as I said, 150 million compounds, where for each such compound we preserve many levels of information. As you can see here, we keep the internal pre-calculated scores from all the point it runs, physiochemical properties, structural information, and compound variants like ionomeric, tautomeric states, and enantiomers. And we also keep the purchasability level, and currently 70% of all our compounds are purchasable. Of course, all this information is updated regularly. So to mine this database in a fruitful manner, we have identified three main categories. These are target-based tags, ag-activity tags, and product compliance tags. And so, for example, the target-based tags are these. These are the ag-activity tags and then the product compliance. And the idea is that we can actually very easily query this database in many, many ways. These are just examples for querying the database. So overall, this compound is very strong, very queryable, modular, and most importantly, scalable, so that at any time we can add many additional compounds. As for the activity modules, in order for a compound to become a product and to be functional, it must comply with a big set of characteristics. And so, as I said, we identify target-based characteristics. In case of herbicides, ag-activity modules and product compliance, and you can see here. Still in light gray, these modules which we are still considering and still are under development, and all of these are in fact machine learning based classifiers which we developed, and the idea is to gather all of these together into multi-parameter optimization. Further concentrating on these ag-activity modules, and this is just an example. In order for an herbicide to be absorbed in a plant, it can be absorbed through the leaf or through the root. And once absorbed in the leaf or the root, then it moves through the plant through the phloem or the xylem. So the phloem and the xylem are vascular tissues that usually conduct sugars and nutrients. And so for these and also for the phytotoxicity, we actually developed machine learning-based classifiers, and the procedure for this development is as follows. First, we gather the tailored data sets, usually from public resources. Then combining with machine learning algorithms, we built the classifier, and then strive to validate it experimentally. And the result of the experimental tests are then fed back into this whole mechanism again. So concentrating further on the phloem mobility, I'm going to show you some initial results. We've done computational proof of concept based on 350 known compounds. And you can see here that we, based on these compounds, we achieved the area under the curve of 0.99. And sensitivity, very high sensitivity and accuracy. And this was validated experimentally, and the results were very very good. And actually, you can see here that it was tested experimentally on, of course, on other compounds, and you can see here the results, and we have achieved. Very well on phloem immobility. So, as for the results, well, currently we tested several targets with multiple 3D models. All of them were homology modeled, and we actually resulted with. The result was 15,000 compounds that were tested in planta, and the success rate was between 2 to 10% per protein's binding side, and this is considered incredible. Just to summarize this part, in order to move from pesticide discovery to drug discovery, all is needed right now is just to replace the ag activity modules here with the ADME-like modules, and this system can be ready for drug discovery, and therefore this opens many opportunities to enter the pharma industry. So I'd like to summarize. Evogene is operating innovative virtual high-throughput pipelines that enable screening more than 150 million molecules in 5 days. We gained lots of experience with targets that have poor structure information, and we developed our in-house ADME-like tools to model in vivo or in planta performance prediction. And with that, I'd like to thank all my colleagues at work from the computational team, from the research, from the chemistry team, from the lab, from greenhouses, and from the field. So thank you very much. Thank you very much for this very interesting talk. Questions?
Okay, I'll just try out. Could you, do you know which step in the pipeline that you showed has the major impact on the enrichment, which is, I understand, is tenfold better than random? Can you estimate or kind of back engineer and to see which one is the crucial step? And I'm asking that because maybe, I don't know, molecular dynamics is not really necessary. Maybe it is the phloem thing that makes the most effect.
Well, yes, it's a very good question. Well, obviously, each one of the steps may reduce the accuracy at the end, but I think we pay very special attention to the modeling and specifically to the binding site. And fix the watermers as much as possible for the later stages. And I guess all the tools that we use along the way, which are actually industrial third-party tools, have their own. They have a long credit. Long been, I don't know if long been in the industry, but state-of-the-art tools. And the persons behind all these platforms do great jobs. That's it.
Biologists are really obsessed with the need to experimentally validate computational predictions. Now you have a double prediction process because you predict the target by homology modeling, and without validating it experimentally, you move on to a compound discovery based on that model. So I wonder, and then at the end of the day, you get a list of, I don't know, 10, 20, 50, what would be leads in medicinal chemistry. So what is the success rate when you put these into field practice? In having actually compounds that are successfully herbicides or insecticides and so on, out of the list of the final hits that you get from the computational process?
Well, I'm not fully sure I understand your question, but as I
wants to know the statistics of the success rate.
Okay.
After 2 rounds of
okay. Actually, okay.
virtual screening,
Okay.
success rate when it comes to relapse?
Well, you know, unfortunately, I wasn't. I couldn't reveal the actual success rate for specific targets. The success rates that I showed are, in fact, the average ones over all the proteins that we have run, and these are quite many. And as you mentioned, we after all this validation, experimental validation process, this is the average success rate that we received over all these proteins. I did not say that, I forgot to say that, mention, but once after the mining, mining by a researcher, we usually end up with, say, thousands of compounds that go to the next efficacy validation steps, and then these go on to the lead optimization, and so I think we end up with, say, hundreds of hits. That's it.
So once you have a lead that was experimentally validated, what's the time course and cost that it takes till it's in the market as a pesticide? Just because I'm from the pharma and I have no idea in your work. So it's interesting.
Yes, okay, this is a very good...
Of course, general ranges of...
Yes, well, okay. Well, the estimation is much less than usually 10 to 14 years in the pharma industry for a single drug. Well, a few years less than that. I think something like 5-6 years. This is the range.
Because you don't have all the preclinical studies in animals and all of that.
Right. The main cost is obviously for the experimental validation by outsourcing, and also for the validation in-house. The validation in-house where we have to purchase the compounds. Right.
Thank you very much.